Yes.
Indonesia tanah air, tanahku harapku. Di sanalah aku berdiri, jadi pandulimu. Indonesia kebangsaanku, bangsa dan tanah air. Marilah kita bersatu, Indonesia bersatu. Hiduplah tanahku, hiduplah negeriku, bangsaku, rakyatku semuanya. Bangunlah jiwanya, bangunlah badannya, untuk Indonesia Raya. Indonesia Raya, merdeka, merdeka, tanahku, negeriku yang ku cinta. Indonesia Raya, merdeka, merdeka, hiduplah Indonesia. Indonesia tanah air, tanahku harapku. Di sanalah aku berdiri, jadi pandulimu. Indonesia kebangsaanku, bangsa dan tanah air. Marilah kita bersatu, Indonesia bersatu. Hiduplah tanahku, hiduplah negeriku, bangsaku, rakyatku semuanya. Bangunlah jiwanya, bangunlah badannya, untuk Indonesia Raya. Indonesia Raya, merdeka, merdeka, tanahku, negeriku yang ku cinta. Indonesia Raya, merdeka, merdeka, hiduplah Indonesia Raya. Indonesia Raya, merdeka, merdeka, tanahku, negeriku yang ku cinta. Indonesia Raya, merdeka, merdeka, hiduplah Indonesia Raya. Atas dasar Pancasila dan Undang-Undang Dasar 45.
Alright, we will start. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Well, good morning, everyone, ladies and gentlemen. The Honorable Head of the Department of Chemical Engineering ITS 2025, Professor Dr. Insinyur Lailatul Qadariah STMT IPM, and also the Head of the Master Chemical Engineering Study Program 2025, Dr. Hakun Wirawasista Amara Marta STMMT PhD. Also, distinguished professor, lecturer, respected guests, and all the participants. Welcome to the guest lecture World Class Professor 2025. With me, Muhammad Habib Tanwirul Ulum, as your Master of Ceremony for today's event. Today is very special day because we are fortunate with the distinguished guest from the National Chung Cheng University, Taiwan, who will share with us about the topic that's very important nowadays. That is Professor Wingyan Michael Chan, PhD. But before we go, allow me to extend our warmest greetings and deepest gratitude to each and every one of you for joining us today. Your presence here is shared commitment and advancing knowledge and building stronger academic collaborations. This is not only learning opportunity for us, but also expand our global academic perspective. For today's topic, that is the compound that can increase anti-cancer immunity. Dear ladies and gentlemen, before we go any further, please rise for the national anthem of Indonesia, followed by ITS hymn. Everyone, please stand up.
Indonesia tanah airku, tanah tumpah darahmu. Di sanalah aku berdiri, jadi pendirimu. Indonesia kebangsaanku, bangsa dan tanah airku. Marilah kita bersatu, Indonesia bersatu. Hiduplah tanahku, hiduplah negeriku, bangsaku, rakyatku semuanya. Bangunlah jiwanya, bangunlah badannya, untuk Indonesia Raya. Indonesia Raya, merdeka, merdeka, anakku, negeriku yang ku cinta. Indonesia Raya, merdeka, merdeka, hiduplah Indonesia Raya. Indonesia Raya, merdeka, merdeka, anakku, negeriku yang ku cinta. Indonesia Raya, merdeka, merdeka, hiduplah Indonesia Raya. Negeriku yang ku cinta, ibu yang memberi desa, dalam bara semangatmu, cita-citamu tak kendur. 10 November 45 mendorong berjiwa ibu, melangkahlah berjuang dengan tekadmu yang teguh. Membina bangsa atas dasar Pancasila dan Undang-Undang Dasar 45 landasan juangmu. Negeriku yang ku cinta, ibu yang memberi desa, langkah kokoh kelenganmu membina putra-putrimu. Menuntut teknologi yang canggih, berbudi anugerah cerdas, menuju kesejahteraan bangsa dan umat manusia.
be seated. And now, ladies and gentlemen, we are moving on the highlight of today's event. That is the guest lecture sessions. This session will be guided by our distinguished moderator, Dr. Hakun Wirawasista Amaramarta, STMMT PhD. Dr. Hakun is highly respected researcher in the field of chemical engineering, with numerous contributions to advanced studies and international collaborations. His work has significantly enriched the academic environment of our department and inspired many young generations to pursue excellence in innovation and scientific inquiry. So without further ado, let us warmly welcome Dr. Hakun to moderate the session and deliver welcoming speech. Everyone, please give him a round of applause.
Okay, thank you very much, Mr. Habib. Ladies and gentlemen, this is our recognized professor in the world, especially in the cancer. You can know the Prof. Vinchan is high index almost 47, professor. Almost 50, almost 50. It's very big high index, So we can learn about the Prof. Chan, take some guest lecture in here. The second, the Prof. Chan became a dean, biological science professor, biological science, and numerous manuscript and reviewer in the top journal, especially in the cancer. So we have some collaboration about the Prof. Chan. So I think enough for the Prof. Chan. Please give applause about Prof. Chan. The title of his speaker is The Role of Epigenetic Modification on the Immunotherapy of Bladder Cancer. Time is yours, Professor. Thank you.
Terima kasih. Thank you very much for the kind introductions and invitation. So this is my fourth time to be in ITS, and it's very nice to visit Professor Hakun, my friend, and also some of the students that I have met before. And it is also my honor to give a talk here. And hopefully today's lectures can help you to understand briefly what epigenetics modifications, its importance, and also the most important is what is the current immunotherapy in cancer, because this is a very important therapy right now. Although you may not have influenza and COVID, both COVID and influenza. So have to take a vaccine. COVID vaccine very important. Okay, and then we have a lot of pineapple. Okay, so this is my university. Oh, there's some echo. Okay, so anyway, next slide please. Okay, now I am doing biology now. Yeah. Can you turn off one of the speaker? Okay, but you turn off this audio, then they can't hear. So just turn off the microphone. I don't know which microphone is turning on. Okay, anyhow. Okay, so as I just asked, no one has taken biology class before, so maybe I need to teach you biology. So there is one very famous person. His name is Gregor Mendel. In 1895, he discovered one very important thing. That is the genetics. So we call him the father of genetics. Did anyone see this term genetics before? No one knows this term genetics. It's so important. So maybe you studied that in a high school. So Gregor Mendel used 7 very important tracts of pear plants like the flower, the color of the flower, the height of the plant. And then what he found out is that there's something inside our human body or inside the living organism to control the phenotype or the characteristic of how the plants look or how we look. So he called it genetics, although he has no idea at that time what it is. For example, we have blood group, right? Blood group A, B, AB, and O. Let me ask you, if one person has blood group A, marry with another person with blood group B, then what will be the possible outcome of the blood group of the kids? So one person has A, the other person is B. So what is the possible outcome? Only AB? A? How about B? How about O? No O? No, you are wrong. So if one person has blood group A, the other person has blood group B, so the possible outcome is can be A, can be B, can be AB, and can be O. That's genetics. That's genetics. So that's the power of genetics. But Gregor Mendel at that time, 1895, he doesn't know what it is. He just knows there is something inside that can control how we look. For example, another example, in African, marry with a Japanese, what does the skin looks like? An African, black, marry with a Japanese, white. And what will the kids looks like? Half black, half white, right? Not very black, not very white, but in the middle, right? That's genetics. That's genetics. Okay. So, but no one knows what it is at that time. Next slide, please. Until a very important discovery in 1953. James Watson and Francis Crick, and also Wilkins and Rosalind Franklin, female. Yeah, okay. She didn't get Nobel Prize at that time. Only 3 persons get Nobel Prize. James Watson, Francis Crick, and Wilkins, they discovered the 3D structure of DNA. Anyone knows what is the name of the 3D structure of the DNA? Anyone has the idea? The name of the 3D structure of the DNA? No one knows. It's called double helix. Have you heard this term? Double helix? This is the X-ray crystallography of the DNA at that time. Because of this X-ray crystallography, they get Nobel Prize in 1962. And then they know that DNA composed of 4 nucleotides: A, T, C, G. You know, you learned that. That's biology. You said you didn't take biology class. How come you know A, T, C, G? Where do you learn it? Ah, you are computational biologist. Oh, you are good. It's very important. I tell you, everyone. So, 2 subjects you have to learn right now. Number 1, AI computer. You have to learn. The second one, immunology. You must know. I will tell you why. Later in my slide, I will tell you why. 2 subjects you must learn right now. Number 1, computer, AI. Number 2, immunology. Okay, why immunology? I will tell you later in my slide. If you read the news, then you will know what I will be talking about. Okay, so Watson and Crick discovered the DNA in 1953, and they get Nobel Prize in medicine and physiology in 1962. Because DNA is so important. Do you know where DNA in our cell is? Where is it located in our cell? Anyone know? DNA, the ACTG. Where can you find the ATCG in the cell? Whatever animal cell, plant cell, insect cell, whatever cell, where is it located? Anyone knows? ATCG, the DNA, where can we find our DNA? Yeah, very good, excellent. In the chromosome, which is located in the nucleus. So DNA, do you know what DNA produce? Eventually produce. No, DNA will produce RNA. This process we call transcription. And then RNA will then produce what? Protein. Yes, exactly. Good, very good. You are good. So what is this process? RNA to protein. What is this name of this process? Anyone knows? DNA to RNA, we call it transcription. And RNA to protein, we call it... Anyone knows? Translation, yeah. Did you check ChatGPT? Did you just check ChatGPT? No? No Google? Okay, good. Translation. So DNA will eventually translate into protein. You know, our whole human body is composed of protein. So how your DNA makes the protein is so important. Different DNA sequence will make different protein. So that's why we have AB, and O blood group. It's because of our DNA. Our skin color, it's from DNA. Whether you are tall, you are short, DNA. So whether you are clever, it's because of your DNA as well. Whether you are fat, you are skinny, it's because of DNA. So everything because of the DNA sequence. So because the DNA sequence is so important that in the US 1995, they launched a very important project which is called Human Genome Project. Everyone heard of this Human Genome Project? So they try to sequence every single DNA ATCG sequence in the human chromosome. So it takes 15 years for them to sequence the whole DNA, our whole DNA. And then now you can find the DNA sequence inside the computer of the National Center for Biotechnology Institute, NCBI. When you go to NCBI, then you can download human DNA or chromosome, the sequence we know. Because the sequence, the combination is so important. It determines whether we are male, we are female, we are skin color, we are tall, we are short, whether we have disease. Because some person may have genetic disease. Maybe when they are born, they have anemia, they have Mediterranean anemia, they have a lot of genetic disease because of the DNA sequence. Now I'm going to tell you a little bit more difficult things. Do I need to stop fixing something? No, I can continue. Okay, so, sorry, previous one. But in 2010, Time Magazine used this photo. Why your DNA isn't your destiny. Time Magazine in 2010 used this photo. So if DNA is so important, why do Time Magazine said DNA isn't your destiny? DNA can make you clever or dummy, tall or short, male or female, whether you have disease, everything. Why? Times Magazine said DNA isn't your destiny. Because there is a small word there. The new science of epigenetics, blah, 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 blah. Now I'm going to tell you what epigenetics, EPI, with genetics. What is it? Next slide, please. I want to make use of this slide to tell you what epigenetic is. So, the first person that discovered epigenetics is Howard Cesar. When he was at Hebrew University at 1977, he discovered the first phenomenon of epigenetics, which is now we call DNA methylation. He is one of the candidates for Nobel Prize in epigenetics, because epigenetics haven't get Nobel Prize. So he is one of the candidates. And I'm honored to host him in 2014 when he visited Taiwan. So he used this slide to tell us what epigenetic is. So, as you know, I've just mentioned, genetics is the DNA sequence, the ATCG. The combination of the ATCG is genetics. So what he said is, by using this slide, he teach people what epigenetics by using this slide. This is ancient Bible. Bible is written by Hebrew. So Hebrew is by Hebrew alphabet, just like English. We have A to Z. Hebrews overall has different alphabet. Different alphabet composed of a Hebrew words. So there are several alphabet and then composed of one Hebrew words. But in the ancient Bible, there is no space between words. So between words and words, no space. Then how do you know where is the word? Very important. By the punctuation of Hebrew, which is underneath the Hebrew alphabet. By looking at the punctuation, you know the words between words. You can separate it. So, what Howard Cesar said is, this alphabet, the combination is the ATCG, is the genetics. And the punctuation is epigenetics, which is on the surface of ATCG, which Gregor Mendel, Francis, James Watsons and Francis Crick didn't know at that time. They have no idea what epigenetic is. So, but Howard Cesar discovered the first epigenetic event, which is the punctuation of the DNA sequence. So, without correct punctuation, the gene can be mixed up, cannot be transcribed and translated into correct protein. So it's very important. So next slide, please. Let me use some illustration to let you understand how important epigenetics is. Okay, so this is, don't go too fast. Going back. Okay. So this is therapist, massage therapist, massage therapist. You don't want this word to be mixed up, okay? Next slide. So you don't want to mix it up this one. Otherwise, no one will want to go into this shop, right? Next slide. This is a US shop. Kids Exchange. Kids Exchange. This is a shop to exchange kids' toy. If you have kids, if you have brother, sister, younger brother, sister, they know, you homes you have toys. Just like now, you have video games in your cell phone, right? So when you play and then you get bored, you need to change a new video games, right? Everyone play video games in your cell phone, right? So, like your younger brother and sister, maybe they have toys. So they don't have your parents may not be allowed them to play cell phone when they are young. So they play toys. So they play toys, they get bored with the toys. So what will they do? Throw it away? No, too wasteful. They go to this shop, exchange toys with other kids. Then the kids can have new toys, right? Next slide. You won't want to mix it up with this word. Kid sex change? No way. I don't want to bring my kids inside and then the sex will change. Maybe some parents want. Yeah, but most of them doesn't want, right? So, this space is the epigenetics. And then this is the ATCG genetics. Do you understand how important is the epigenetics is? If you have the wrong epigenetics mark and the words cannot be correctly translated. Next slide, please. So, what is epigenetics? Epigenetics is, as I first mentioned, what Howard Cedar discovered is the DNA methylation, which is the first epigenetics phenomenon, which is on the surface of the DNA, which have some methylation. You are chemical engineering, you know what is methylation, okay? It's a CH3 group, so you know. And so we have several other epigenetic modifications, which doesn't change the DNA sequence, but change the chromosome or chromatin structure. So it will change whether the genes can be expressed or not. That is epigenetics. Next slide. So one of the phenomena which I will be talking about today is called DNA methylation. So DNA methylation occurs at the cytosine. This is cytosine, chemical structure. Did you learn this chemical structure before? So have a CH3 group attached to it. This is normal phenomenon. Normal, okay, by an enzyme. And the enzyme uses some substrate, which is taken from your food as adenosine methionine. It is from your nutrient, from your food, as substrate, and transfer the CH3 group by this enzyme to this cytosine. It will become 5-methylcytosine. But interestingly, in living organism, methylation only occurs at the cytosine, which is followed by G, by guanine. So, methylation only occurs at the C of CG, but not in standalone C. You understand what I say? Methylation only occurs at the C, which is followed by G, but not in the standalone C, which is CA, no methylation. CT, no methylation. CC, no methylation. Only CG have methylation. Very interesting. And we have a lot of CG, which is cluster in the certain region of a gene, and it controls whether one gene can be expressed or not. And if a genes get methylated, transcription will not occur. DNA cannot transcribe into RNA. Next slide. So let me tell you how important is DNA methylation. So do you know what it is? Honeybee, right? So you know honeybee. You also know, I guess you also know, we have queen and you will have worker. You know, right? So the queen hiding inside the beehives. Okay? But do you know that DNA sequence is the same? Do you know that DNA sequence is the same? Because they are developed from this worm, they either turn into queen or either turn into worker. So whether it go into queen or workers depends on what it was fed. If it was fed by worker jelly, which you don't want to take, it will become worker all lifelong. But if this worm was fed with royal jelly, you know, do you have royal jelly in Indonesia? It's very expensive one. We have royal jelly in Taiwan, which is very expensive. So this worm will become queen. So what it fed will change its life completely into either queen or worker. Why? Do you know what paper it is? Science? Do you know this journal? No one knows what science is. Do you know there is a two prestigious journal, Science and Nature? Have you heard? Science. Oh, thank you. So, do you know Science? Which country published Science? Which country published Nature? Anyone knows? Did you read Science and Nature paper? No? Too difficult to read? Very short. Usually, Science and Nature paper is very short. Do you know the impact factor of science? 40 or 50. Yeah, if you publish a paper in science, you get famous. Maybe you will get Nobel Prize if you can publish a paper in science. So science and nature is the most two important papers in natural science. So nature published by the British and science published by the United States. If you want very novel study, read nature and science. You'll find it the most important, the most novel papers, novel research. So in 2008, Science publishes paper said that nutrition control of reproductive status in honeybees while what? What is this? DNA methylation. So what it is, change the DNA methylations of the bees and it becomes different honeybee, whether it is queen or worker. So that's why it is so important when you, especially when you are young. So you must have the correct food, not junk food. You must have the correct food in order for you to develop properly. If you don't have correct food, the correct means balanced diet. You have carbohydrate, you have protein, you have lipids, you have everything, you have vitamins, everything. A balanced diet, you will develop correct DNA methylation pattern in your body, and your genes can be correctly transcribed and translated. If you have malnutrition, you do not have enough or correct food, you don't have the proper nutrient, the DNA methylation cannot be developed correctly. So, you'll become worker all whole life, your whole life, you become worker. So very important. Next slide. So that's why right now we understand we are developed from one single cell. Our human body are developed from one single cell, right? Sperm and egg, one single cell, which is called embryonic stem cell. And then it developed into our whole body, right? So our body, from your head to your feet, the DNA is exactly the same. From your head to your feet, all your cells have the same DNA. Why? We have different tissue. Because different tissue have different DNA methylation pattern, have different epigenetics modification. So our body uses epigenetics modifications to develop our cells into different lineage in cellular differentiation. So important. It's so important. So your body don't want to mess up the epigenetics. Otherwise, it will become different things. Not different human, but the human with some defect. For example, next slide. For example, because of the genetic changes, ATCG sequence, because you eat a lot of junk food, so junk food has carcinogen, which change your DNA, and also improper epigenetics mark, such as the DNA methylation is not correct. it will result in the inactivation of the tumor suppressor gene and the activation of oncogene, and eventually it become tumor. So tumor is because of the inactivation of tumor suppressor gene. We have two kinds of genes in our cell. One is called tumor suppressor gene, the other is called oncogene. So because of the genetics alteration, changes in genetic sequence, changes in ATCG, because of what? Because of carcinogen. So where can you find carcinogen? Smoking. So you eat a lot of preserved food, smoking, preserved food, alcohol. Maybe, I don't know if you have beetles in Indonesia. So in Taiwan we have lots of beetles. The beetles, beetle nuts, the beetles. So you have oral cancer. Taiwan has a huge number of oral cancer because they chew the beetle's nut. And alcohol or virus. So we have hepatitis B, hepatitis C virus. It will change our DNA sequence, and the person with hepatitis HBV or HCV infection will have liver cancer. The liver cancer is because of virus that change the DNA sequence, or because of epigenetics change, improper DNA methylation that occur at the tumor suppressor gene, shut it down. So, what is cancer? Why it's come from? It's because cancer is from the accumulation of alteration of genetics and epigenetics changes, result in the inactivation of tumor suppressor gene and activation of oncogene. So that is cancer. Now we know everything on cancer. We are very clear of what cancer is. Let me say it again. What is cancer? Cancer is developed from genetics and epigenetics events, and it accumulates in cells. So you will not get cancer in one day, but you may get cancer in 10 years because the carcinogen will accumulate in your body. You smoke one cigarette, you won't get cancer. But you smoke one day, one cigarette, you may get lung cancer in 10 years. So that's carcinogen. And epigenetic changes. So carcinogen will also induce epigenetic changes, will make DNA methylation occurring at the tumor suppressor gene. So altogether, cancer is because of the inactivation of tumor suppressor gene and activation of oncogene, and making our cell become cancer. So that's cancerous. Next slide. Okay, let me make you give you a more complex view of what cancer is. So, in the past, we always, in the treatment of cancer, we only focus on the cancer cell. But it all fails. Because in an actual tumor, it's not only composed of cancer cells. You can look at this figure. In the actual human tumors, it composes of tumor cells and also blood vessel, and the most important is composed of a lot of immune cells. So we have two types of immune cells. One is anti-tumor, so for example the natural killer cell NK cell, which I will talk about. And then we have T cells, which I will also talk about. But we also have some immune cell which is pro-tumor. Pro-tumor means it helps tumors. So, for example, this one I will talk about in the next slide, the Treg cell or regulatory T cells. So, you can see this, the tumor is so complicated. If you only focus, if your treatment only focus on the tumor cell, it usually fails. Because we have a lot of different cells which help tumors to regrow. So if you only focus on the tumor cells, the immune cells, the pro-tumor immune cells will help the tumor cells to regrow again. That's why in the current most advanced cancer therapy, it is not focusing on the tumor cell. It's focusing on the surrounding immune cell. So altogether, we call this tumor microenvironment, because the actual tumors, composed of cancer and non-cancer cell. So right now we know the non-cancer cell is more important than the tumor cell itself, because the non-tumor cell support the growth of the cancer cell in this whole tumor. So that's why during the drug development, we must think of that. If you only think of the tumor, it's failed. In the past 50 years, we all know it's failed, because we only focus on the tumor cell. We must develop drugs that can also focus on the immune cell as well. Next slide. Let me tell you how important this is. Anyone read news yesterday night? Yesterday night announced the 2025 Nobel Prize in Medicine. Anyone read this news? Please raise your hand. It's announced at 6 p.m. Indonesian time. I read news. I always follow. So don't play your TV games, okay? Read news. Tonight, 6 p.m., we'll announce the Nobel Prize in what? Anyone knows? No one knows. Tonight we'll announce what? Tonight. So for first one, Nobel Prize, always medicine first. And then the second one is what? Chemistry. And then physics. And then economics. And then later will be the Peace Prize. The Peace Prize. So the US President is one of the nominees for the Nobel Peace Prize. Do you think he will get it? Donald Trump. Do you think he will get Nobel Peace Prize? I don't know. Okay, go back to this. Yesterday night announced 2025 Nobel Prize in Medicine. Who get it? 3 scientists. But the most important one is Shimon Sakaguchi from Osaka University, Japan. I met him several times. I listened to his talk several times. He discovered the regulatory T cells. Can you go back to the previous slide? So regulatory T cell, or we call it Treg cell, is one of the cells in our human body that impose a break in the immune system. It's so important. The regulatory T cells is the one of the immune cell in our body that impose a break. Without regulatory T cell, what will happen? So the person will develop autoimmune disease. Have you heard of autoimmune disease? SLE, for example, SLE is one of the autoimmune disease, very common in women. Or we have the rheumatoid, so which is also autoimmune disease. That is, the immune cell attack your own body. So type 1 diabetes is autoimmune disease. Type 1 diabetes, not type 2. Type 2 is because of you eat a lot. But type 1 is because of autoimmune disease. That's your immune cell, your T cell, attack your pancreas. Attack the beta cell of the pancreas. So your pancreas cannot produce insulin. So that person will develop diabetes when they are young, very young. That's autoimmune disease. So regulatory T cell is a key cells that control our immune system, not making our immune system too strong. So if after your inflammation, if you have virus infection, your immune system are activated. So when after the clearance of all the virus, for example, COVID, if at the early 2020, before we have the vaccine, there's a very high death rate of COVID. Why? Because it's not because the virus kill you, it's because the inflammation kill you. It's not the virus kill you. Because the inflammation, because the virus trigger an inflammatory response, a very huge inflammation response, and that inflammation response, that immune response kill you. So before the vaccine, that's because of the inflammation, the immune response. And the response is so strong that even the regulatory T cell cannot suppress it. But usually, when you have virus infection, say influenza or common cold, so you have fever and you get cough, and after you get this, you get the virus, your immune system are triggered to kill the virus. But after that, regulatory T cell will suppress your immune system and then back to normal. So when we talk about immune system, we always talk about balance. Balance. You have on, you also have off. So very important, we have balance in our immune system. Balance. We have on and we have off. And then tumor is so clever to activate regulatory T cell to suppress the T cell. So suppress the T cell. So why do I talk about that? Next slide. And the next slide. Next slide. And I want to talk about that in 2018. We have the Nobel Prize in Medicine to another very important immune regulation, to James Allison and Tsuku Honjo. So this is Professor Tsuku Honjo. I took a picture with him in 2019 when I attended the Japanese Cancer Association annual meeting in Kyoto. So Professor Honjo discovered PD-1 and Professor James Allison discovered CTLA-4. What are they? Next slide. CTLA-4 and PD-1 are two important proteins on the surface of T cell. So T cell supposed to be able to kill tumor. Our T cell can kill tumor. Our own immune system can kill tumors. But how come tumor derive? How come tumor growth? If T cell can kill tumor, then why we have tumor? Because the tumor is clever. Tumor express the ligand of PD-1 and the ligand of CTLA-4. What are they? They are the brick of T cell. The brick, like your car. You have accelerator and you have brake. Like regulatory T cell. Regulatory T cell also a brick of the immune system. But this one is the protein of the T cell which serve as the brick. So when you press it, the T cell will stop growing. The tumor is so clever. They use PD-1 ligand, which is called PDL1, and CTLA-4 ligand, which is called B7, to activate the PD-1 and CTLA-4 so that the T cell stop growing. The T cell will stop, kill the tumor cell. So because of this discovery, right now we have two very important FDA-approved cancer drugs, which is an antibody. So chemical engineering student, you should be able to engineer protein. So if you study protein engineering, then you know how to synthesize this drugs. So anti-PD-1 and anti-CTLA-4 is right now very important drugs in the cancer immuno or cancer immunotherapy. We call this immune checkpoint blockade, ICB, immune checkpoint blockade. All over the world, patients right now are using immune checkpoint blockade. This drug doesn't kill cancer directly, but this drug, they break the bond between the PD-L1 and PD-1 or B7 and CTLA-4 so that the T cell can kill tumor. So it uses T cell to kill tumor. It's very successful, very important. So that's why after the launch of immunotherapy of immune checkpoint blockade, people think tumor can be completely erased because we remobilize the immune cell to kill tumor cells. And the tumor, as I mentioned, is so clever. It also can mobilize regulatory T cell to suppress T cell. That's why Shimon Sakaguchi get Nobel Prize last night because of this important discovery that the regulatory T cell will also be the target of cancer therapy. Okay, next slide. So if you want to know more about what immune checkpoint blockade is, Google Jim Allison breakthrough and read and watch this documentary. So this documentary by James Allison tells you how he discovered the immune checkpoint and how he pushed this discovery into the actual clinical market. And they also interviewed the first patient that received this antibody drugs. And that patient has melanoma. And that melanoma was first being treated with chemotherapy, but not successful. She thinks she will die. But because she entered the first clinical trial of this drug, so that patient get completely cured. Melanoma goes away, never come back because of this very important. So you can Google and you can watch it free. I don't know whether Indonesia can Google this breakthrough. And then James Allison plays saxophone here. So when I attend the cancer meeting in the United States, he has a band. He has a band. He has a band. The band is called the checkpoint because the protein that he discovered is called checkpoint protein. So he named his band the Checkpoint. So he played saxophone here, over here. And then all playing here, they are all MD or PhD. Here, everyone, they are either MD or they are either MD, PhD. So they are all scientists. They all play, they make a band. So this is from the MD Anderson Cancer Center of the University of Texas, one of the most famous cancer institute in the world. So he is from that institute. So you can look at that documentary and then you will know how important is immune checkpoint inhibitor is. Next slide. So now I'm talking about my own research work. So I work on bladder cancer or urothelial carcinoma. So why? Because it's one of the most important cancer in Taiwan, and its prevalence in where I located because of probably arsenic contamination. So beware of arsenic. So it will make people have bladder cancer. The incidence rate of bladder cancer in Taiwan is 8.03 per 100,000 in male and 2.82 in female. This incidence is likely higher than Indonesia. So 6.3 in male and 4.0 in female. Female rate is higher than Taiwan. And bladder cancer ranked 13 in Indonesia. So we rank number 8 in Taiwan. And in Indonesia is 13. I don't know what is the number 1. You may check. Next slide. So bladder cancer is very painful. Although the death rate is not high, but it's painful because you need to have a cystoscopy to go through the urethra and then to take a look on whether your bladder has a tumor. So that's the diagnosis. So it's very painful. Next slide. For the treatment, right now, besides we have the regular chemotherapy, and now people are using immune checkpoint inhibitors. As I mentioned, immune checkpoint discovered by James Allison and Tsuku Honjo. So, but the success rate is only 20%. Very sad, only 20%. Next slide. Why? Because for the patients that are responsive to immune checkpoint inhibitors, it has a lot of T cell, a lot of immune cell, a lot of good immune cell in the tumor. If the tumor doesn't have good immune cells, or it has no immune cells, or it has only bad immune cells, it will not respond. So this treatment is so important, very effective, but only effective in around 20% of the patient. And I tell you, it is expensive. How expensive? So one complete course is 1 million NT dollars. 1 million Taiwanese dollar. If you want to treat, 1 million Taiwanese dollar. So how much equivalent to Indonesia? 1 million NT. 30 million. 300 or 30. 500 millions Indonesian dollar. So in Indonesia, you must have this therapy, but only very rich people can use. But in Taiwan, right now, our national health insurance pay for it. Patient doesn't need to pay. Our Taiwan Taiwan have national health insurance, so you pay every month. You pay very low, and then you can use the health insurance. Taiwanese government pay for it, but not everyone success. So it will waste money for the patients, not success, right? So we want to help all patients can be successful. So why? Because the patient doesn't have good immune cell or to have no immune cells. So everyone in my lab, including in my lab, is thinking how to make this kind of tumor, I call it cold tumor. It's cold tumor because it doesn't have immune cells. So it's very cold. Make it become hot. A lot of immune cells. So make it from cold to hot is the most important oncology question right now. If you can make a tumor from a cold tumor into hot tumor, then the immune checkpoint inhibitor will be successful. So how to make it? Including my lab found that epigenetics is the way to go. Because this tumor has the wrong epigenetics mark. So the immune cell cannot go, or only bad immune cells go. Only regulatory T cells go. So that's why we have to turn this into this. If we can turn this into this, then the immune checkpoint blockade can be successful. Our government won't waste money. And everyone, maybe everyone in Indonesia, everyone can use this therapy at no price. Okay, next slide. So what the drugs that I am using is called cyproheptadine. Cyproheptadine. What is cyproheptadine? Cyproheptadine is the first generation antihistamine. Antihistamine is if you have allergen after you eat shrimp. Maybe you are itchy. I'm so itchy after I eat shrimp. Some people, when they eat shrimp, they itchy, right? Then you have to take antihistamine. Cyproheptadine is the first generation. First generation means 50 years ago it was launched. Very safe drugs, but only one side effect. You take one, you won't wake up for 3 days. No, I'm kidding. So you can wake up, but you feel drowsy. I can't work because I'm so sleepy. That's the side effect of cyproheptadine. Anyone take cyproheptadine before? You ever take cyproheptadine? I take it before. Oh, very strong. But the itchy go away immediately almost. You don't feel itchy anymore, but you feel sleepy. Okay, so there is one patient with liver cancer. The liver cancer already spread to lung. Spread to lung, very serious. Last stage, they have only 3 months survival. After 3 months, the patient will die. And then the patients get itchy because he take the targeted therapy, thalidomide. There's anti-angiogenesis, suppress the blood vessel. And so the doctor think, oh, he get itchy and he is the end stage liver cancer. Maybe I subscribe him with cyproheptadine. We have second generation and third generation antihistamine, which we will not feel sleepy. But the doctor decide to prescribe him with cyproheptadine because he want to make him sleep so that he can pass away peacefully. But after 3 months, next slide, tumors gone completely. No tumors at all. And then the patients get healed. Whoa, how come? So there's another case, similar case, and also it's the same. So he published a paper in BMJ Case Report, which is a very good journal in the UK, in the United Kingdom. Unexpected remission of hepatocellular carcinoma with lung metastasis to the combination therapy of thalidomide and cyproheptadine of 2 cases. Wow, it all gone. How come? Why cyproheptadine do it? Next slide. So this is cyproheptadine. Looks like very simple structure. So the pharmacologist told me this structure make you sleepy. This one. So the third and the second and third generation, they change this structure. So make it won't feel sleepy. So now you can prescribe fexofenadine, which is the third generation antihistamine. So, but I tell you, the effect is not as good as this one. The anti-itching effect is not as good as this one. This is the best. So fexofenamide, it's okay. You will feel a little bit itchy. You have to take more. You have to take more. Take 1 week until you don't feel itchy. But you won't feel sleepy. So which one you want? You want this one? Immediate itchy go away, but you feel sleepy. But you take 1 week drugs, but you won't feel sleepy. Which one you want? Maybe all people like the second one, right? They don't like the first one. The first one, I feel sleepy, I can't work, I can't drive. Yeah, but some people take it as sleeping pill. They can't sleep at night. So they take 1/4. No, don't take 1 pill. If you want to sleep, 1/4 is enough for you to have a good sleep. 1/4. Yeah. Okay. So it's from 1950. It's developed. So it's very cheap. $1 NT per pill. In Taiwan, just $1 NT. $1 NT. $1 Taiwanese dollar. which is $500, 530 in Indonesian dollar. Is it cheap? 530 Indonesian dollar. Is it cheap? It's very cheap, right? Okay, it's very good one. It's still on the market. Next slide. So that scientists, that doctors come to me and ask me, Michael, do you want to try if cyproheptadine is useful in bladder cancer? Why not just try? I try it. This is the cell line study, MTT. MTT. You see? This one, the white one, is the normal cell. The normal cell, no effect on cyproheptadine. It won't kill cyproheptadine. It won't kill normal cell. But cancer cell die. Very safe. It's very safe. So cyproheptadine will not kill normal cells, just cancer cell. So when you develop a drug, remember, it must be non-toxic to normal cells and toxic to cancer cell only. Very important. Next slide. I test animal. This is the tumor in the animal, and this is the control, no drug. This is the tumor. This is the mice taking the cyproheptadine to tumors become very small. So not only cell line, but animal study also important. Next slide. So we perform sequencing, the next generation sequencing, to ask why. Did anyone heard of next generation sequencing? No one heard of it? Then go to biology department. So yesterday night, I talked with a professor on, she studied insects and ecology. There's another one. She is the director of the undergraduate study. Another ladies in biology sit next to Professor Avi. Yeah, she is the one that she knows how to use this. You can ask her. Okay, next generation sequencing. So I perform next generation sequencing to examine why cyproheptadine is useful. Next slide, next slide. Next slide. I found that, natural killer cell mediated cytotoxicity. Next slide. It is enriched. Wow, after cyproheptadine treatment. So this pathway, NK cell probably being triggered. Immune cells, good immune cells. Next slide. So then I want to study why. Why? Is it really the immune cell? So, so I teach you about the T cell before. Regulatory T cell and T cell. So Nobel Prize, right? Yesterday night Nobel Prize is to regulatory T cell. Yeah, so now remember, every October, watch the news for Nobel Prize. Don't watch K-drama. Okay? No K-drama in October. Okay? October, always Nobel Prize. Not get Nobel Prize first. Not K-drama, okay? So, very important. You have to see which one get Nobel Prize. And then study. Oh, what is the discovery? What's so important? Why that person get Nobel Prize? So, biology, always the number one is medicine. And then physics and then chemistry, right? Okay, so next slide. NK cell is one of the cells in our human body. It's the first line of immune system. It can kill not only tumor but also virus, bacteria. Very important. And NK cell can recognize tumor cell by this receptor on the tumor, which we call the NKG2D ligand. So NKG2D is the receptor on the NK cell that can sense bad guy. So bad guy, this is bad guy, right? Tumor is bad guy. So NK cell is good guy. So good guy has NKG2D. Bad guys has this one. So but why police cannot capture bad guys all the time? Can police always capture bad guy? Sometimes no, they escape, right? Like here, next slide. Because the bad guy hide the NKG2 ligand, they hide it, so the police cannot find it. Wow, the tumor is so clever. The tumor is clever, escape the police, escape the NK cell. Why? Why it shut down? Why it disappear? Because of epigenetics. The tumor use epigenetics to hide it. So you have to make it re-express again. So let's see whether I can re-express it. Next slide. So I use a co-culture system. So it's cell culture. So you can learn in my lab if you want to do this experiment. It's difficult. So you have the cancer cell. I label the cancer cell with fluorescent color. So this cancer cell has color, fluorescent. And this is natural killer cell. So I treat this cancer cell with the cyproheptadine or not. Control is no treatment, and then treated is by cyproheptadine. And then I co-culture it again, and then see what happened. So if the cells are killed, the fluorescent will be released into the medium, released outside. And then so you measure the fluorescent signal, and then you know whether the cancer cells are being killed or not. Next slide. So next one. Next one. Next one. Okay, here. Okay, so let me teach you how to read this graph. The red one is the cell treated with cyproheptadine. The white one, white bar, no treatment or control treatment with DMSO. Okay, so we treated with DMSO or cyproheptadine. This number is the ratio between the NK cell and the tumor cell. This one here, no NK cell, only tumor cells. And this is the rate of the death, the death rate of tumor as measured by the fluorescent amount. So you can see here, without the NK cell, the tumor will not die. Tumor is not dying. Tumor is very happy without NK cell. But when you add NK cell, the tumor start dying. But if the tumor cell is cultured with treated with CPH, it die more, right? The graph is show higher. More NK cell kill more tumor cell. And then this is significant. P-value less than 0.05. Have you ever studied statistics? And do you study statistics? Did you use T-cell, T-test, student T-test? Do you know how to use student T-test? No, you don't know statistics. So you have the mathematics department in the Faculty of Science, right? Go to take some statistics class. You must know statistics because you work on computer, right? You know some statistics, right? A little bit? Okay, did everyone hear about T-test? Anyone know T-test before? They didn't learn. So maybe you have to take statistics. Very important. Very important statistics. So why important? Because not only for this one. If you study statistics, you can go to the bank to work. The bank use statistics to calculate the stock market, everything. Very important. Okay? So you can see treatment after CPH, the tumor cell get killed. Next slide. Next one. Okay. So next one, I skip this one. It's the same by using, okay, this is interesting. So let you see how, so here, this is the tumor cell, and this is the NK cell. You can actually see the NK cell kill the tumor, right? So this is treated with the compound, and this is not treated. And then you can kill much faster. So it is 7 hours. So this is a time-lapse microscope, 3D microscope. So 8 hours, 10 hours is a fast-forward mode. 10 hours, 11 hours, 12 hours, 13 hours. Oh, tumor's gone. So much faster than this one. You can see it at the very beginning it has more NK cell, and this NK cell coming is slow, right? NK cells coming slow coming, but for this one treated, NK cell keep going and kill it. So the police can find the thief, the bad guy. Good guy can find the bad guy much faster, and more good guys go, more police go to capture the thief. So this is good. Okay, next slide. Okay, so why the good guy can find the bad guy? Because ULBB2 or the NKG2 ligand are re-expressed after treatment. So the identity restored. Why it's restored? Next slide, next slide. Because we restore, next one, next one, next one. Okay, we restore the epigenetics. We change the epigenetics of the tumor cell. The tumor cell's epigenetics change. We didn't kill the tumor. We just changed its epigenetics so that the good guy, the police, the NK can find the tumor and kill it. So I didn't kill the tumor. If I kill the tumor may knows I want to kill it. So it will change the tumor. It will change, it will develop some mechanism to escape my drugs. I didn't kill it. Okay, NK cell kill it. Next slide. So this is cell line. Cell line is not good. We have to do animal. So animal, actual, in vivo. Next one. So we inject the tumor into the animal. Without treatment, the tumor grows very fast and large. But after treatment, it grows slow. Okay? It suppresses the growth. Next one. So because, okay, this is the tumor weight, smaller weight. Next one. And it has more NK cell because NK cell get in and kill the tumor. Really NK cell, yes, we do the staining. We take the tumor and we dissect it. We send it to the hospital. And in the hospital, the pathologist help us to stain the tumor. And then this is the brown one is the NK cell. We can actually see the NK cell in the tumor. So NK cell kill tumor. Not only in the cell culture, but also animal. Okay, next one. So next one, next one, next one, next one, next one. Oh, sorry, go back. So why? Because we found that the tumor can also excrete a cytokine, which is called CCL3, which attract the NK cell to them. So the tumor not only changed the identity, but also call the NK cell, come in, catch me if you can. Catch me. So they send CCL3, the tumor sends CCL3 and tell the police, I am here, come here to catch me. So the tumor becomes stupid because of the cyproheptadine. Probably maybe, oh, I know why, because the tumor gets sleepy after the cyproheptadine treatment. Yeah, cyproheptadine make it sleepy. So it doesn't know, it already tells the NK to come. Next one. So what my research tells is this, is because originally, so the tumor use epigenetics to shut down the NKG2D ligand, so only very few NK cells to kill tumor. Next one. But after we treat the cell with the epigenetics drugs, it's reproduce the NKG2 ligands and also CCL3, and so the tumor has more NK cell to kill it. So we restore the immune tumor microenvironment. So good immune cell go to the tumor. Okay, next one. So this one is just published in July of United Issue of Molecular Medicine, Impact Factor 6.4. So by two graduate students, Himani, my PhD student from India, and Nina is a Taiwanese student. So they are the first two author. They share the co-authorship. So I am here. Okay, next one. And so during my study, there is another paper published in Cancer Cell. Cancer Cell is a very important cancer journal. Impact Factor 40. Another Impact Factor 40 paper. So they found that fexofenadine, which is the third generation cyproheptadine, also has the same effect as cyproheptadine to kill the tumor cell. So when it suppresses the allergy, so the T cell can restore the killing. What they found is not our finding is NK cell kill tumor. What they find is T cell kills tumor by using antihistamine. So fexofenadine is the third generation drug. So it will not make it sleepy. So allergy is not good. If you always have allergy or inflammation, it will help tumors grow. So don't make yourself allergy, okay? So next one. So summary. Cyproheptadine inhibit bladder cancer cells, but not immortals bladder urothelium in vitro and in vivo. So what we found is epigenetic therapy may be able to enhance tumor immunology, immunotherapy, while epigenetics priming of immune-related genes. Okay, so next one. Okay, so this is a review that I wrote in 2020, that in epigenetics inhibitors is very important to restore the attack of the T cell to tumor cells and suppress the effects of regulatory T cell, which Nobel Prize yesterday night, right? And also another T helper cell. So epigenetics inhibitor right now is very important. So it's one of the FDA approved drugs right now. Okay, so I think I stop here and then let you ask questions before I go to introduce the TIP program again. Okay.
Thank you very much, Professor, for inserting the presentation. Do you have some question, guys? Have some question? Mr. Fuad, do you have some question? Okay.
Sorry, can you repeat your question? Any epigenetics treatment on what? In different cancer. Yes, immunotherapy is not only effective on bladder cancer, it's effective of a lot of cancer right now. So not only bladder, so it's on a lot of cancer. So liver cancer also, yes. So when you ask the doctor, you ask oncologist, what kind of cancer can be treated by immunotherapy? A lot. Yes. Very important therapy, but expensive. Yeah. Maybe you don't understand at all because you have zero biology background, or you understand something. Anyone understand something? Please raise your hand. no one
Chemia
understands.
organic. Do you have
So
Okay.
anyone completely zero, I don't understand what you say. I don't completely understand. I understand zero. Raise your hand if you understand zero.
I think we have chemical organic, Professor. The same with this
Okay,
one.
okay, okay.
I think we can.
So that means you all understand 100%.
No. It's very interesting, Professor, about the structure of compound, the natural product with the epigenetic. Because if we want to uh, decrease the cancer, so we can take epigenetic. Is it have some the same with structure in the natural product? About
For
the
hyperteptidine, I don't think so. But there are lots of natural products that can be epigenetics, that have epigenetic function. There are lots. For example, green tea. The green tea already known to have epigenetics effect. So green tea can also enhance immunotherapy. It's been known. So drink more green tea, and a lot green tea. Vegetables also, yeah. Vegetables are already found to have epigenetic function as well. Eat more vegetable. Yeah, so eat less barbecue. Or before you eat barbecue, eat vegetable first. Fresh green leaves has a lot of epigenetics, good epigenetics compound inside.
Have some about the structure. The structure must have like a panel or something to take get epigenetic effect, Professor.
Yes, there are some structures. So, so for example, can you go back? Go back to the slide with the video. Oh, here, yes. Okay, you can see this one. This is a very important epigenetics inhibitor. It has this similar structure. If you can find a similar structure as this one, then it can be a good epigenetics drug. Yes. Yeah.
Thank you very much, Professor.
No problem. Yeah.
Do you have some question, guys? It's very interesting. Yeah. Okay. Yeah.
Yeah, very good question and important question. So, actually, it's very difficult for me to comment on that because I don't know the treatment of your grandfather. So, but basically, so why the tumor comes back again? It's because the drugs cannot totally kill the tumor, because there are some tumor which is drug resistant, so it's hiding. So the regular chemotherapy cannot kill all the tumor cells. It kills most of the tumor cells, but not all. So the one that are not being killed has the properties which is very similar to stem cell. I don't know whether you heard of the term stem cell. So stem cell is a very important cell. It can differentiate into everything. So anyway, so this cell has a properties of drug resistance. It resists. It cannot be killed by normal drugs. So, but these cells will hide, and then after 3 years, 5 years, they will then start growing again. And then when it starts growing, it will be different because it's drug resistant. You cannot be killed by that drugs. That's why people propose not using the drugs directly to kill the cancer, but to mobilize the immune system to kill the cancer. So why it is useful? Because if our immune system are being mobilized, it can monitor. It helps you to monitor whether the cancer is there or not. So it will suppress. Even it come back, it's suppressed. But if we use chemotherapy, most of the time, all the immune cells are killed. The chemotherapy kills the good immune cell as well. So there's no immune cell to help you to monitor where the tumor is. So that's the reason of why chemotherapy at the very beginning it seems to be successful. But after 3 years, 5 years, it comes back because some of the tumor cells they hide and they didn't come back. They did come back after 3 years, 5 years. That's the reason. Yes. So that's why we are now focusing on the tumor microenvironment. Very important this term, because we are not in the past 10 or 20 years, in the past 20 years, we only think of cancer cells. That's incorrect. That's right now we know it's totally incorrect. Because in the tumor, in the real tumor, not only cancer cells, there are immune cells, there are blood cells, there are lots of cells there. You have to think of how to use those cells to kill tumor, not directly kill tumors.
I think we have. Good. You have some question again, guys? If you can, okay.
How you find the active compound to kill the cancer cell. Okay, so right now I would suggest if 5 years ago or 2 years ago, I would say just do MTT and screen everyone. But right now I would say use AI. So use AI to do the structure prediction right now. So because you probably has a lot of compound. So if you have a library of compound, you can perform a AI modeling and to predict which one will target what protein in the tumor. And then, because now we have a lot of AI prediction software, such as AlphaFold. You have AlphaFold, so which do you know the last year Nobel Prize in chemistry and physics? Do you know? Last year Nobel Prize in chemistry is the one that developed AlphaFold, the chemistry. So it's AI. And physics also AI. So last year, 2 Nobel Prize, physics and chemistry, give to AI people because they use AI to predict. And then physics is for AI because we need physics to build a better computer for AI. So they give AI the Nobel Prize last year. So that's why are you using ChatGPT every day? I am using ChatGPT every day because of the physics, because of the chemistry. Everyone use AI every day because now you have for chemistry people, so you have the AlphaFold. You can use the AI modeling to predict what is the target of your structure. After you get the prediction, then you do experiment. It saves time, saves money. So we are not doing the screening right now by experiment. It wastes time, wastes money. Do AI first, and then select the best one, and then do experiment. It saves your money, and then faster. Yeah. Everyone is doing it in Taiwan right now, doing AI search first.
Oh, it's good treatment to save the money, Professor.
Save money, save
Yes,
time, save
save
money.
time,
And then
yes.
after AI selected the best one for you. Yeah, and then you get better result faster.
The validation of the AI is almost 100%, Professor.
Of course not, but it's better than nothing, right? It's not 100%, but it screen out a lot of junk. If something that AI think is not useful, then you can just kick it out. And then you just select the one that AI said is useful. But you still need to do experiment. I read the papers, they make it much, much faster to do clinical research. From the drugs to human, now it can shorten the time. Shorten it very much. Because before it takes 10 years. From discovery, do cell line, and then do animal, and then do human, 10 years. Now it's only 5 years. And then it saves money, because before you have to test 100 drugs. Now you only test 10 drugs, or even 2 drugs. So it's very important. You have to study AI. You have to learn how to use the chemical tools, because you are from chemical engineering. You must learn how to use the AI modeling. There are lots of good AI modeling for that. Otherwise, you have to write it yourself. So programming, very important. You are young, you can learn programming. We are old, we cannot learn. They can learn. I always ask my student, do this one AI. So in my lab, I have 2 teams. One doing experiment, one do computer. So they do all computers. So we do experiment and send it to the computer people. And then they do computer calculation, AI modeling, and send back to us. Okay, do this gene, do this protein. So I have 2 teams. So I need to buy a very expensive computer. So your computer must have 256 giga RAM. So 256 giga, your RAM. Yeah. So 8G is not enough, 16 not enough, at least 64. Your computer have at least 64G. So computers, our computational sciences lab, in CCU, our computer science lab, their computer is 256G. And CCU is going to buy one supercomputer right now from NVIDIA. So they're buying the GPU, from the NVIDIA guy, Jensen Huang. Yeah, he's very famous. He always wear the black suits, right? Jensen Huang.
It's very, very good treatment, Professor. I think our publication have 2 publication in AI, Professor. Already
Your
2000.
your
Yeah.
Because yesterday, Professor Arvid from biology said they collaborated with
Yes.
mathematics. I think your mathematics department should have the
Yeah.
AI.
My student, Badril,
Yeah.
have a good AI.
Good.
Yeah.
Yes,
So,
important, very important.
Yeah, so can take collaboration with the
Yes.
AI,
Yes.
chemical engineering AI.
Must do.
Yes, thank you, Professor. Have some question? So you must learn about AI, right? It's very important, yeah. In now because of the big data. Yeah, the big data must have the
So, can you go
After-
back? Can you go back? Go back. Go back. Go back. Go back. This is, they use AI to calculate this one. So, because the data is too many, too big, so my team used AI for calculation. So I didn't show you how, but it's the calculation. Yeah. You must have computer team to help you. Yeah, but students are smart. They learn fast. They can do it.
You have some question, guys? Enough for this one? So, enough? So, Professor,
To explain, to introduce
about
the
TM, yes. TIP.
Okay, go.
We have
and
a
then-
TIP program, yes, if you want go to Taiwan. Okay. That is your Professor.
Yes. Okay. Go. Next one. Okay. So maybe you want to go to Taiwan to join my conference. I'll be organizing an epigenomics symposium. If you want to learn more epigenomics, come to Taiwan. So we have a poster, whatever. So next one, we have scientists from. Next one. We have scientists, speakers from Hong Kong, Japan, Thailand, India, something like that. Okay, next one. Okay, now I introduce you two things. First, the STEM program, a PhD program in my university. STEM stands for Science, Technology, Environment, and Mathematics. Next one. So this is an English taught PhD program. So you can take any of this subject: physics, chemistry, chemical engineering, biomedicine, environmental science, mathematics, interdisciplinary training. And then we collaborated with the industry as well. Next one. So this is the subject that we have. So the student can join any of the subject for PhD. Okay, next one. So for that one, we have scholarship. So don't worry about that. If you apply, guaranteed you get scholarship. Enough for you to live very good life. Yeah. Coming to Chiayi and going to Taipei is different, because Taipei is expensive. Chiayi is cheap, so much cheaper. So you can eat very good, sleep very good in Chiayi, but in Taipei expensive. Okay, the other one I want to introduce you is TIP, Taiwan Experience Education Program. So if you want to come to Taiwan for 3 to 6 months, you can join this program. So this is our government internship program. So everything is free. It's free. Next one. So we pay for dormitory, your living, and air tickets. Everything you pay. So don't worry, you just apply. But you have to be associated with Professor Hakun, because so many students apply. I cannot accept everyone. I only accept my friends, student. Yeah, so if you want to come, you have to get into Professor Hakun's lab. Otherwise, you can go to others' lab. I mean, in CCU, there are so many professors accepting the tip. If you want to go to my lab, I only accept Professor Hakun's students. Yeah, so, but if you go to other lab in CCU, you can apply. So you just search TIP and CCU and you can find. Next one. So we last year, because this year not finished, so we have Indonesian, Philippines, Thailand, Vietnam, India, and Malaysia joining the TIP program. So next one. So this is the ITS student in my lab before. So this is Rizky, your student, right? And this is Chandra. And this is, I forgot her name. Lindsay, yes, Lindsay. So they came to my lab as TIP student before. Yeah, so this is other TIP student. You can meet other TIP students. So this is from Thailand, Chulalongkorn. So you can meet with them. Thailand, Chulalongkorn, how many? One, and so this is one. And then here, okay, this is the last one. Otherwise, I have another 3 or 4. Okay, next one. So if you join our STEM program, if you come for PhD, then you can maybe you are able to work in our semiconductor industry. Have you heard of TSMC? Do you know what TSMC, Taiwan Semiconductor Manufacturing Company? So anyone heard of TSMC? No? No one do know what TSMC is? Anyone use iPhone? No one use iPhone, too expensive, right? iPhone, the chips inside is from TSMC. So right now, 95% of the chip is manufactured by Taiwan TSMC. So in the world, in the whole world, over 95% of the chip is manufactured by TSMC. The other 5% by Intel or by Samsung. So China a little bit. China is a little bit. If you are using the China cell phone, probably is a if you are using Huawei anyone use Huawei cell phone? Is using TSMC chip, Taiwan chip as well. So Huawei is also from Taiwan. The chips is also from Taiwan. So if you want to go to work in TSMC, you can take our semiconductor curriculum offered by TSMC. So next slide. So you have to take 11 courses out of those courses. So out of those courses, some of you, if you are chemical engineering, it's easy. So for example, analytical chemistry. Do you have analytical chemistry? Probably, right? So you take 3 out of this 10. So, and then 5 in this 20. So for example, we have chemical engineering class. No problem for you, right? Organic chemistry, no problem. Physical chemistry, no problem, right? Then it's easy for you. And then you have take 3. You have take 3. So one very interesting one is called semiconductor practice. Okay, next one. What is semiconductor practice? You can go to TSMC, Newcomer Training Center, to play around with all the machines that are involved in semiconductor manufacturing. So you play around with very expensive machine. So it's a 2 weeks course. You don't need to pay, you just need to join the curriculum and then you can go. So 2 weeks and you learn everything. And then, next one. So after that, you will get a certificate of TSMC. TSMC will give you a certificate. And with this certificate, what you can do, next one. So, with this certificate, next one. Sorry, back one. With this certificate, so if you get hired by TSMC, your salary will be very high. It's very high. Higher than me. Higher than me, even higher than me. Because in Taiwan, working in semiconductor, you get very high salary. How high? The first year, guaranteed. The year and whole year, your salary more than 1 million Taiwanese dollar. 1 year. The first year, your annual salary will more than 1 million NT dollars. So it's very high, higher than assistant professor, more than an assistant professor in Taiwan. Yeah, so that's if you want to work in TSMC, then you have to join this program. So not only in CCU, but in other 13 national universities in Taiwan, including the NTUST. NTUST also has this program. So if you go to NTUST, they also have this TSMC semiconductor curriculum. Yeah. Okay. So next one and the end. Okay. So any question on the TIP or a STEM program you want to come to Taiwan to study? Short term, long term? Or you want to come to Taiwan to work in semiconductor?
Okay. You have some question about the TEF or the semiconductor. It's a very famous in Taiwan, yeah. The bigger salary in Taiwan in the semiconductor plant.
It's high salary, high tech company. I went to the TSMC headquarters before. Wow, you want to work inside because it's high class. Their office, their building is high class. They have Starbucks inside. The Starbucks inside their building, in the canteen, they have McDonald's, they have Starbucks, everything good restaurant because their R&D building has 3,000 people working in the R&D. And at the underground, they have a very large canteen, including Starbucks. But you cannot bring cell phone inside. When you get in, you have to pass through the X-ray machine. No computer, no cell phone, no iPad. Every electronics stay outside. So very high tech. You want to work inside.
Do you have some question about the challenging this one? Okay.
Okay. So for the undergraduate student, you can come as TEP, T-E-P. But for TEP, you cannot join the TSMC program. The TSMC program is only for degree student, for undergraduate, master, or PhD. So not for TEP. TEP you cannot join. Yeah. The TEP program? Okay, the TEP program can be as short as 2 months to 6 months. So, but I would say longer is better because 2 months you cannot learn something because there is a ITS biology student just left and then come back here. He stay only 2 months, not enough. Yeah, I met him yesterday in biology. Yeah, he just came back from CCU. He stayed there from 2 months. And there is another student from ITS biology. She is now staying in the lab doing experiment. Yeah, she will be staying for 6 months. Yeah. Yes.
Professor, I want to ask about TSMC. Is for the graduate student, magister have a degree of master,
Yes.
and after that can take this program.
So as long as the student is degree student, is okay.
So not graduate from the master is okay.
Master okay. Master, undergraduate, master, PhD can take.
Okay.
It's for degree student, not only in CCU but all over Taiwan,
Okay.
also NTUST. So if you want to take this program, you must be a degree student. Undergraduate, master, or PhD is okay.
Maybe you want to apply or something. It's okay. Yeah. But TEP you can take in the Prof Chan lab or some the other lab professor. It's okay. Yeah. Because international exposure is very good for you. Yeah. So you can learn about how to the habit of research in Taiwan.
So the chemical engineering department of CCU also accept TIP student. So not only biology, not only biomedical science, you just search TEEP and CCU and you'll find all the professors from our university that accept TIP student. You can apply. But because there are so many applicants, it's better you have a professor that knows CCU professor. Then it can help you. Okay, accept my student, please. Otherwise, it's difficult because so many every year. We have 200 to 300 TIP students from Thailand, Indonesia, Philippines, Malaysia, India. So you can see in the map. So many come to CCU. So we don't know whether we should accept you or not, because I don't know whether you are good or not. But if you have a recommendation letter from professor here in ITS that know someone in CCU, it will be better.
Enough, guys, or you have some question? Yes. This. What can,
Okay, you will do research work. So we will join in the research work of the lab's own research. Or sometimes you can bring your project if you have collaboration. So you can bring your project also. Yeah.
Enough, guys? Or do you have some question again? Yes, too many. Yeah, almost 8 or 10. Yeah, 10 student of chemical engineering, professor, go to the program in the Prof Chan laboratory and the other labs. So you can take this challenge, yeah, to make a good one, yeah, a better one, because some people now is Safrina, professor. Yeah. Safrina have a good performance in the MTT essay, so she become a lecturer in the Brawijaya University. Only take 4 until 6 months and qualified with the MTT essay. This one. Okay. Okay. Doesn't have again. Enough. I think if you don't have some question, so maybe give applause with the Prof Chan. Thank you very much, Professor, for interesting topic and interesting presentation. Thank you very much. We have some a good insight, yeah, because different insight for our research is a good one, Professor. So I think can become a complexity to make solve the problem. Yeah. Okay, I think enough. Thank you very much for your Professor Chan. I think time is back to MC.
All right. One more, give the round of applause for the Professor Chan, also for Dr. Hakun. And now session, it's the Dr. Hakun will give the present for the Prof Chan. So the documentary. Right, still there more. Oh yeah, sure. All right, maybe one more with all the participant, Professor. Take a picture with all the participants. Yeah. Maybe all the participants can go. All right, let's go. thank you so much. You can go back to the seat. All right, what an insightful and inspiring session we have today. So, ladies and gentlemen, we have gained so many valuable perspective from our distinguished guest, Professor Chen from National Chun Chen University, Taiwan. Let's give one more round of applause to all the professor here. Our sincere appreciation also goes to Dr. Hakun for exactly moderating the session, and of course to all the professor, lecturer, and all participants. It's been a pleasure seeing your energy, curiosity, and passion for learning throughout the event. May today's discussions not only broaden our understanding, but also inspire further collaboration and research among us. Before we close, I would like to remind you that this program will continue tomorrow, so don't forget to joining also for tomorrow. So make sure to join us again. We're looking forward to see there tomorrow afternoon. Once again, thank you so much for your participation and attention. So I am Habib as your master of ceremony for this event. It's really honor for accompany you throughout this event. And lastly, wassalamualaikum warahmatullahi wabarakatuh, and see you tomorrow.